As you can see in this chart, the HIV proteins that affect pathogenesis include TAT, VPR, NEF, VIFIF, VPU. The TAT is transcriptional transactivator. It accelerates HIV transcription by host polymerase. It's also secreted from the host cells, and upon entering an uninfected cell, it triggers apoptosis, or programmed cell death. The VPR viral protein R regulates the import of HIV into the nucleus before the viral cDNA integrates into the host chromosome. The NEF, or negative factor, downregulates CD4 and MHC class I proteins, protecting the infected cell from killing by CD8 cytotoxic T cells, slows apoptosis in infected T cells. Secreted NEF activates apoptosis in uninfected lymphocytes also. VIF, or virion infectivity factor, counteracts a host cellular protein that naturally inhibits HIV replication, and VPU, or viral protein U, counteracts a host cellular protein that inhibits virus assembly and export. Case study, sex warts and cancer. Janine is a 45-year-old migrant crop worker from Belize. Over the past 2 months, she's noticed abnormal vaginal bleeding, accompanied by a foul-smelling yellowish vaginal discharge. She was also experiencing increasing pain in her pelvis. She visited a free clinic near the California farm where she worked, hoping a simple medicine would help. The clinic doctor performed a pelvic exam and colonoscopy to view her cervix. Colposcopy, to view the cervix. He saw an advanced stage of cervical cancer. He took a sample for DNA testing and a pap smear to confirm the neoplasm. Janine admitted having 6 sexual partners over the years. She also recalled seeing a wart-like growth in her genital area about 20 years ago. At the time, she was too embarrassed to go to the doctor, but the warts disappeared after a few months, so she thought nothing of it. A week later, the pap smear results confirmed his diagnosis, late-stage cervical cancer. The DNA test revealed the presence of human papillomavirus DNA. Tragically, Janine would never return to Belize. So Janine had not received routine pap smears and was sexually active. She noticed a small wart-like growth in her genital area, but never sought treatment. She was diagnosed with late-stage cervical cancer, and the test revealed human papillomavirus DNA. So this is an example also of human papillomavirus actually on somebody's fingers. So it can get to many different places of your body. In the non-disease state, normally PRB neutralizes the ETF, a protein that's needed to make essential cell division proteins. E2F stimulates cell division. In the disease state, HPV viral protein E7 chaperones the human protein PRB towards degradation. Then the proteasome's degradation of the PRB begin, and this leads to uncontrolled cell division. Human herpes viruses and latency. After a primary infection, the human herpes viruses become latent in the host cells, the nerve cells or white blood cells. And this is kind of similar to what we talked about before with lysogeny in the bacteriophages. The DNA circularizes and exists as an episome. Then the DNA integrates into the host cell genome. The latent virus can reemerge after years of latency and cause a new active infection. Small RNA molecules called microRNAs made by herpes virus interfere with the host cell's apoptosis program. Okay, let's talk about protozoan pathogenesis. We'll describe different ways protozoans invade host cells. We'll correlate the immune response to protozoan-induced damage to the host tissues, and explain several mechanisms the protozoans use to avoid the immune system. So, immune avoidance by the protozoa. Antigenic masking. In this case, some protozoans coat themselves in host antigens to avoid detection by the immune system. Also, antigenic variation. As you can see in this example, you actually have changes in the antigen that cause them to be seen as a totally different organism. Here we see the trypanosome that's coated with VSG. One cell switched to blue VSG. The green VSG protozoa are killed, but the blue VSG cells repopulate the blood. Intracellular location. Immunosuppression. Some protozoans induce secretion of anti-inflammatory cytokines to reduce the immune response. I'll get out of this. It requires another login before I can watch the videos because it's not coming directly out of my Moodle site. So I will get out of that for now. But make sure you go in and you look at any of the animations that go with Chapter 18. And also make sure you go through these clicker questions yourself when you have time. But I'll go ahead and pop them up here and read through them anyway. In which way is a genomic island different from the rest of an organism's chromosomal DNA? And the answer is D. Genomic islands have a different percentage of guanine cytosine base pairs than chromosomal DNA. Which of the following is a bacterial virulence factor? A. Capsules. Which of the following is a property of a bacterial exotoxin? C. It's made of protein. Which of the following bacteria would produce endotoxin? The answer is E. coli. It's a gram-negative.